final lecture of this session. That's me. First of all, I want to thank the organizers for organizing this wonderful meeting. And I'm glad I'm speaking on the second day because I've collected the questions in the past 24 hours and I integrated some of it in what I'm going to talk here today. So this is what I mentioned at the beginning. We're in the broadband moment and we can change the fate of not only epigenetics but also genetics. I'm going to talk about genetics and genomics throughout life at very early stages where mutations are extremely critical, during the lifespan when we have to decide which drug to take, and also genetic is important in the afterlife, and I'll mention that at the end of the lecture. I'm going to tell you 3 stories from our lab, and I'm going to tell you 3 rules of how to establish or send out ideas from your lab and materialize them in companies. So we work on genomics like many of you, and I call it a triangle. We combine biology, computational biology, and clinical questions, and all together we have to reach better decisions next to the bedside. I think we heard that excellent in the previous lectures. So many of us take a group A and B, so it could be before disease, after disease, it could be a time course, it could be before drug and after drug. And if we identify the difference between the two, we can call it a biomarker. If we understand it, we can understand the mechanism. And if we understand the mechanism, we can offer therapeutics. And we're using tools of reading, understanding, and writing. And again, we heard that from Eric Green yesterday. Reading has increased drastically in the past 20 years. And we're getting better at understanding. We heard that in the previous lectures. And we can also write it and modify it and change it. When I need to give examples of how much we advanced in the past 20 or 30 years, I say that the first human genome was equivalent to 4 jumbo jet airplanes, and today it's the cost of a skateboard. And that's the leap between 4 jumbo jet airplanes and any skateboard we can buy today. And as mentioned, we also use the Ultima Genomics platform, and we compare it to Element Biosciences, we compare it to Illumina, and we compare it to the other methods as well. If you think about it, we take all this biology and it becomes digital on our computer, and then we ask our computer to think a little bit, like a human being using AI, and turn it back into biology. So this is our cycle, collecting a lot of biology, it's digital, and then trying to explain the digital and getting back to biology. I think we all agree, and again, we've seen this before, we have to be proactive and not reactive as early as possible. That would be best. The 3 stories from my lab are going to be how we identify genetic diseases very early during pregnancy, how we stop the spread of breast cancer in mice, and how we better assist trauma and forensics. Genetics throughout life. Let's start at the beginning of life, where mutations determine the difference between life and death. So our bodies built out of billions of cells, and we have our genome inside every one of our cells. In our blood, we have circulating cell-free DNA, roughly at the size of about 150 nucleotides. During pregnancy, the ratio is 9:1. 9 pieces of mother's DNA, 1 piece of embryonic DNA. That's in the first trimester. So week 10, 11, and 12, the ratio is 9:1. During pregnancy, in order to make sure that pregnancy occurs or the baby is intact, you can use sonography and also invasive tests such as amniocentesis or CVS with a risk for the pregnancy. In 2011, a test came out called NIPT, non-invasive prenatal testing, and in this test, you actually count the amount of DNA in the mother's blood. It's a mixture of mother and embryo, and if there's a tiny bit more of chromosome 21, the mother is not sick. That means that the extra chromosome came from the embryo. This is like looking at planet Earth from outer space. Is there another North America? Okay, that's the question. But what if we want to see every nucleotide in the DNA? Can we double-click on planet Earth and look at the continent, and look at the state, at the city, at the street, and even the house? And this is what we did. So we took the same blood samples from the first trimester, And then we wanted to separate them based on features. So this is a real graph. Here in red, you can see the fragment size of the embryonic DNA in the mother's blood. And in black, you can see the mother's DNA in the mother's blood. You can see that it's slightly different. When we started adding additional features, so fragmentomics, we heard about that yesterday, looking at the GC content, where it came from, and other features, and we used Bayesian statistics, we were able to separate it much better. Eventually, we used the AI trick, we used a deep learning algorithm, and we were able to completely separate the embryonic DNA from the mother's DNA. And a few years ago, we sequenced the entire embryonic DNA from beginning to end, fragments of about 150 nucleotides in size, obviously billions of them, just by separating them better. We had a huge bag of mother's DNA, a smaller bag of embryonic DNA, and we were able to resequence it from beginning to end in a simple blood test at week 10. We were also able to do it at week 7, and we believe if we look at the karyotype, we'll be able to do it also at week 5. I want to show you some data here. So this is fetal fraction, the fetal fraction versus the different genetics inherited. So you can see from left to right, and at the x-axis you can see the 5, 10, and 15% fetal fraction. This is roughly in the first trimester. If we look at the paternal one, which are easiest to identify in the mother's blood, you can see even at 5% our performance is well, and at 10% and 15% our performance is almost perfect. Again, if we look at single nucleotide variants here, and we can see different inheritance, we can see different genetics here. This is about analysis of 16 families. You can see the coverage of the parents is at 30x, and the cell-free DNA, about 10% is embryonic. We run at about 300x. And you can see here the performance. At the bottom, you can see our Bayesian analysis algorithm on the left-hand side, and when we add machine learning and when we add the deep learning, all of them gets better, and we really reach very high performance. I want you to very carefully look at these two images. They are almost identical. Can you see the difference? Can you notice the difference between the two? Okay, each one of these images has about 1 million pixels, and I removed 1 pixel. So as you can see, it's not that critical for the image. Our DNA has 3 billion pixels in it. Anybody willing to give away a pixel from their DNA? Come on, 1 pixel. Okay, nobody. Well, you're right. You don't know where that pixel will land. That means when we look at images, we can always, reduce the size. We can remove pixels. When we look at our human DNA, we need the 3 billion pixels because many of them could be critically or could have a critical effect. However, if I ask a very simple question, is there an image there? About 40 pixels are enough to answer that question. But if we want to see every nucleotide and every pixel, this is what we see on the right-hand side. If you think about it, this is the genetic test we do today, the prenatal testing we do. We're looking at chromosomes or part of chromosomes. And what we really want to see is every nucleotide in the DNA. Is it critical to see every nucleotide in the DNA? Well, 1% of all pregnancies carry a disordered related mutation, and if we take all the current genetic tests, we'll see only about 10% of them. We know that the US market is changing drastically, and early identification is very important, and it aligns values of parents, OBGYNs, geneticists, governments, and payers. It's actually a crossroad of the fact that there's a great reduction in the cost of sequencing, genetic information is increasing, the average parent age is increasing, and the average number of children per family is decreasing as well. So it's comprehensive, it's very early, and it's risk-free because it's a blood test. We have the blood from week 10, and we look at genetics, but we can also look at epigenetics. So we did our same trick. We took the blood samples at week 10, we put it in our freezers, we followed thousands of women, and at the end of the pregnancy, some of them suffered from preeclampsia, some of them from GDM and other diseases. We go back to the freezers, we take the sample from week 10, and we profile non-coding RNAs in the blood in order to identify preeclampsia. So we took 40 future preeclampsia, where the blood sample is taken at week 10, and 40 controls. we were able to predict preeclampsia based on 25 non-coding RNAs in the blood in a simple blood test in the first trimester. The advantage of identifying it early is that you can prevent preeclampsia by administering aspirin at low dose if you start it before week 16. So this is the performance here. So the bioinformaticians in the lab were quite disappointed. You can see the random set is in red and the performance is in green here. However, when we discuss it with physicians, you don't always need close to 100 sensitivity and specificity. In this case, all it means is that you administer aspirin at an early week, and you can prevent most cases. And we compared it between samples from the first trimester and the second trimester, and we can see that analysis in the first trimester is still better than the second trimester, meaning that it might be early stages of changes of non-coding RNA that can affect, we have shown, hypoxia, angiogenesis, and other pathways as well. In order to confirm that this was not based on a microbiome change, we swabbed the placentas of preeclampsia and non-preeclampsia, and we see that the microbiome and virome is similar between preeclampsia and non-preeclampsia, so it's not a microbiome or virome effect. Yesterday someone mentioned, why don't we start even earlier? So we have a project with Hadassah Hospital where we take the seminal fluid and follicular fluid and we profile molecular events between these two, the seminal fluid and molecular fluid, before the IVF process. After the IVF process, we follow the pregnancy, then we have successful and non-successful. We go back to the freezers, we take out the extracellular vessels, and we profiled, and we believe we can be much smarter now in understanding successful versus non-successful IVFs. So we talked about early development. What about during lifetime, and how can we stop the spread of breast cancer? Very often we sequence genomes from beginning to end, whole genome sequencing. We do it very often for rare genetic diseases in order to find the causative mutation of this clinical manifestation. And you can see the black dots. So we have many mutations. We want to reach the black dots, and we combine various filters. Obviously, we want to look at the coding region that we can explain, and we want to see how frequent it is in the population, and we want to reach the black dot where we have novel mutations that are critical or drastical. So the left-hand side is severity, the right-hand side is rarity, and we have additional features as well. But what if we want to reach the red dot? What if we want to reach actually sites which are non-coding and happen to be prevalent in the population, not that rare, and can also affect a phenotype? So having that in mind, we started working also on microRNAs. Just as an example, microRNAs are the nails that get stuck in the mRNA bus. Okay? Tiny snippets in our cell that can stop the expression of mRNA just by binding to them and send them for degradation. So crossing data sets. We took a data set of microRNA targets, so mRNAs that have their microRNA targets on them, a database of SNPs and mutations, and sets of genes of interest. So here in the center, we will reach sites for microRNAs that have polymorphism that might affect a set of genes that we're interested in. And we wanted to run our first test on breast cancer due to it prevalent in the population, and we wanted to target the metastatic effect and not the primary tumor. So this is crossing the data sets. We have breast cancer genes, those that have a microRNA binding site with high fidelity, those that have SNPs or mutations in them, and those that affect the cytoskeleton organization. Okay, so here we reach a set of a small number of genes, 19 genes. We picked one of them for our testing, and that is paladin. Paladin is a cytoskeleton-associated protein, which is critical and very important for the movement of cells from one site to the other, the metastatic process. We ran many experiments in vitro with different cell lines, and this is the final experiment we have carried out. This experiment was carried out in mice. I just want to tell you that paladin has 2 binding sites for 2 microRNAs, highly conserved in the population, and very interestingly, the polymorphisms in some populations are about 50%. So in Ashkenazi Jewish, 50% would have a G, the other 50% would have a C, some would bind and some would not. This is the experiment we carried out in mice. Triple-negative breast cancer induced in mice at day 0 by injecting into the mammary fat pad. About 2 weeks later, removal of the tumor and waiting 20 days later to see whether there's any metastatic sites. And in the final experiment, 2 days before the operation, we injected a nanoparticle. So this is a nanoparticle trick where we have gold nanoparticles with a green color that we can follow, slowly releasing microRNAs and controls and cisplatin, so they were complex, and I'll show you the final experiment. In short here, no drug means no cisplatin, with drug means with cisplatin, and you can see the 2 microRNAs that bind pallidin. The tumor is in red, the nanoparticles are in green. And this is the final image here now. So here you can see the nanoparticles injected 2 days before removal of the tumor on the left-hand side, which release a control RNA. Everywhere there is a red arrow, that's a metastatic site in the lungs derived from the breast. This is the control. You can see many metastatic sites, and this is the experiments. Between 0 to 1 metastatic sites, when our gel slowly released these 2 microRNAs that shut down pallidin and a few other genes as well, and prevented the moving from the breast to the lungs. So it's a genetic and epigenetic effect, we call it, because pallidin, we know, could evade the binding by changing one nucleotide, and we also know that the cells play around with the levels of the microRNA. It's either upregulated or downregulated, so you can play with a regulator and play with a binding site. Another experiment we carried out in the lab with exactly the same cells was injecting them IV intravenously or injecting them orthotopically under the mammary fat pad. So the ones on top have a bonus, okay, they're right in the blood cells, and the other ones are in the mammary fat pads. At the top you'll get metastatic sites after days or weeks. At the bottom, after weeks or months, you get metastatic sites. Obviously, at every point, we profile the microRNAs, we profile the mRNAs, and we look at them, and the one that stood out is miR96, meaning that overexpression of miR96 really reduced the movement of the cells from one side to the other, and what the cells do in order to remove or release themselves from the primary tumor is reduce the levels of miR96, and then they can move from one side to the other. We show that in animals, and we also look for correlation in humans. On the right-hand side, you can see the Kaplan-Meier plot with the levels of miR96 and various levels or stages of breast cancer. We also want to do it rapidly, or we want to see if we could use the Oxford nanopore technology, and we heard a little bit about that yesterday. But instead of classical bioinformatics and translating it into letters, we translate it into a signal, and we train our system to look at the signal, how a cancer cell looks like and how a non-cancer cell looks like. And here, in this example here, we were able to separate genomic DNA from mitochondrial DNA just by training the system to see how both DNAs look like, and in 20 to 40 minutes we can separate the 2 DNAs. Here I'm just showing you how easy it is to use the nanopore. I have a course where everybody sequences feces that I get from the zoo, and their final assignment is to identify which zoo or which cage I took the feces from, and they have to identify just by sequencing in the class. We're also sitting outside the operation rooms where we receive fluids, saline, which from the internal cavities of the body. We run the nanopore in 20 to 40 minutes in order to see if we can identify reminiscence of cells before the operation is over, to give a feedback or advice to the physicians, you know, whether there are cells or not, and whether they should continue the operation. So it's informative, it's rapid, and it's smarter. We don't only profile DNA, but we also profile RNAs. Now, there's a few spin-off companies from my lab, and we apply it. It's great because you feel you could get closer to the bedside, not only academic, and I summarize it in 3 points. What do you have to do? And I tell this to my students. What do you have to do in order to spin out a company from your lab? The first rule is to say what this lady is singing. What does she sing? Let it go. Exactly. As a scientist, you have to understand that you have to let it go and transfer it to industry. The other thing is what Mufasa told his son, Simba, and that's remember who you are, okay? Remember you are the scientist. You don't know everything. And if you get stuck, you always have to remember that. You have to keep on swimming, of course. And if you follow these 3 rules, all this will be yours. How we better assist trauma and forensics. So we have a long-lasting study with Professor Talma Hendler for more than 10 years where we collect samples of recruited soldiers at 3 time points: when they reach the base for the first time, during their training, and after their training. Now, these are healthy individuals at the age of 18. We know that at least 10% of them will suffer from PTSD, so we have the samples throughout. They undergo fMRI, and we profile the RNAs in their blood, and we believe we have a predisposition for PTSD very early in development, for early intervention. We do the same with microbiomes, so we have a potential signal from microbiome, swabs from the mouth, and correlation to PTSD, and we work on the active material of cannabis, THC, at a very low dose, 1/10 the weight. We see that it increases cognition, and now we're testing the effects on PTSD and MDD in mice. The advantage, obviously, is we can sacrifice the mice and look at the molecular effect, but we're also linking it to changes in RNA in the blood of patients. So it's data, it's critical data for early intervention, and this is what we're profiling. In a dynamic world where a phone is not only a phone, a watch doesn't only show you the time, we know that medicine changes, and I think we heard it here a lot in the past few lectures. Genetics throughout life, so we talked about before, during, and what about afterlife? This is the lab, half bioinformatics, half experimental, the best in the world in trying. We try a lot, we succeed a little. But our lab has changed drastically, and our life in Israel has changed drastically since October 7th. Many of the students are in Army Reserve, have been in Army Reserve. Many of the other students are volunteers with refuge children that had to leave their homes. I say we always have more than one role in Israel. My day job is at the university. My night job is as a forensic scientist in the Army. So I help establish the unit, and in October 7th, since then I'm in Army Reserve, and we receive various samples and try to match the samples from the field to DNA left behind. I also have an early morning job that's with the family, and an afternoon job that's with the various companies that came out of the lab. So what do we do with forensics? In forensics, you look at short tandem repeats in the DNA. You look at about 23 or 24 sites. And it looks roughly like that, and you can see the various peaks. If you look at about 20 or 24 sites, STR sites in the DNA, the chances of finding another match on planet Earth is 1 to a few billion. However, the problem was, in the past war, is that a lot of the DNA was partial, low quality, low quantity, multiple DNA one on top of each other, and at various stages after exposure to toxins. So upon recruitment of a soldier, usually you collect 3 sets of information. You have the fingerprints, you have the dental images, and you have the DNA. In order to identify a victim, you have to identify 2 out of the 3. However, in the past war, due to the various conditions, that means that at least 50% of the samples had to undergo analysis using DNA and not the other 2 features. Every soldier recruited in the past 20 years leave their DNA on the Watman paper you can see there. However, some of them are older and some of them have not left their DNA, and not all of them were soldiers. So in order to identify the AM, the ante-mortem, to the PM, the post-mortem, a lot of analysis was carried out on samples received from biopsies taken and left in the hospitals, from corona swabs left in the hospitals as well, and 31 women were identified using the pap smears they underwent in the previous year. So you have the PM that you find in the field. In order to match it to the AM, you have to find the DNA left behind. Now, these are the real numbers. I'm showing it to you here because it will be published soon. This is the identification based on DNA only in the first year of the war, since October 7th, 2023 to 2024. You can see direct events, about 2,000, about 60% of the events. And then you have the highly complex one, forensic investigation, and complex mass fertility incidents, where you have more than one identification and you have to separate them, and we're using computational tools for that as well. So how do we define success? We know success when you identify a mutation and you know the phenotype, when you find a set of mutations and you can predict a potential drug that can help it. How do you find success in forensics? Well, you define success in forensics when you identify a victim. Now, why is that success? That is success because the alternative, we know, if you don't know, is much worse than that. Nathan Alterman wrote a poem in 1936 about three mothers that sent their soldiers, their sons, to the war. Two of them have a very vivid image of their sons, and one of them does not. The first mother says, "Onia me'alechet bayam hasheket ubni talui al rosh hatoren." There's a ship in the ocean, and I see my son at the head of the mast. The second mother says, "Bni gadol v'shatkan, hu olech basot, hu yagia ad kan, hu noseh belibo kadur oferet." I see my son, he walks in the field, he has a bullet in his heart. And the third mother, which is the one that suffers most, doesn't know where her son is. "Va'em השלישית בעיני התורה לא היה לי יקר כמוהו, איכה אבך לקראתו ואינני רואה, אינני יודעת איפה הוא." אז הבכי רוחץ את ריסיה שלה ואולי הוא לא נח, ואולי הוא מודד בנשיקות, כנזיר משולח את נתיב עולמך אלוהי. I cannot see my son, he's walking in the fields, he's walking in the route towards God. This is a word in Hebrew. It's very short. It's dafka. If I have to explain it or translate it to English, it's despite everything we have to keep on going. I think we have that in mind in Israel. Everything we undergone, and with the help of the US now, things are getting a little bit better. I think we have to keep on going. I think the science will keep us together. Genetics throughout life at different stages, 3 stories, 3 rules. In the past 2 years, I noticed that I started writing many editorials about the best, worst job in the world, seeking the soil. Scientists should be the buffer of turmoil. There's a lot of bad news outside. I think if we speak a lot about science, we know that many things good may occur. Don't devoid emotions of science, and 2 other ones that are now in publication, War-Life Balance and 90 Seconds Nation, how in Israel in the past 2 years, everywhere you are, anytime, any day, you are 90 seconds away from shelter. You are always waiting for a siren. Our lab supports diversity. Our lab supports art and science. Stay hungry, stay foolish. Thank you.